Right. So this really is providing at an undergraduate and graduate school level. But I realized that you could do a short version of this, which is called "How to Get a PhD in 1 hour." But really this is an introduction to experimental design for you all, and just an attempt to summarize what I've learned over the last few years. So experiments really are there to generate new knowledge, and knowledge as such is actually a field. It's called epistemology, but knowledge is debated by philosophers and so on and so forth. But in really pragmatic terms, if you can find the information that you seek, right, in the literature, Google, PubMed, or our own lab archives, our data vault, you really already have the knowledge you need. And I think it's a practical kind of first quest. Is the question I have really something that requires an experiment, right? So new knowledge is really needed for problem solutions in our world of medicine and science. So without data and clinical information, you really can't have any true progress. You can practice what you know, but you really don't learn anything new or improve. And if you look around, you always see that, or I see that exploration is a motivation. Whether you are an explorer or anybody who has curiosity about the world, you will try to develop new ideas and concepts. But really, experimental design, as Caleb asked me, at least to my mind, is a professional approach, right? It's a job, to be humble, to get relevant data and provide an objective result to generate new knowledge. And there's a lot of words there, but data and objective is something you need to think about. So I'll give you a little bit of my view of the history of how science generates new knowledge. And I'm particularly interested in old and new school, right? Because as I mentioned previously, it's really about observations. And there is sort of a struggle between making good observations and having what the old guys or old scientists used to call observational logic, and then make it into something objective, right? And there is history on this that goes back to India, that goes back to China, to Middle East, Europe, and so on and so forth. But for our today's very brief introduction here, what you typically see around people discussing knowledge is something where they make an observation and then try to find out cause and effect, right? They deduce something, or by inductive logic, they have some kind of facts that they make fit into a system. And my favorite battle here in this kind of generation of new knowledge is sort of Aristotle, that became our favorite Italian here, Galileo. So Aristotle was really famous at his time because he was able to write up conclusive logical systems that explained everything. The earth was flat. You know, if you if you looked at the horizon, you'd see that at one point you have to fall off. And seriously, this is what people believed. So they had fantasies about some kind of hell and heaven where somebody would fall off the edge of something, because that's how they observed the world, right? Now, in Europe with the Renaissance, Galileo, of course, thanks to a little bit of mathematics, we have Copernicus and so on, but Galileo, I think, embodies this battle. So he made observations, right? He had telescopes, and he decided based on shadows of the moon, which were of course round, that either it was a disk or a sphere, and he presented this. But it was interesting because the logic of that was then conceptually different. And people really told him, even the church told him, that because they had read Aristotle, even though they were religious, there was no other truth that needed to be added because all knowledge was already known, right? And he went for house arrest, of course, etc. But he fought the battle versus accepted and versus new. And you get new by empirical. And by empirical, it's sort of a lot of terminology here, but empirical means something that you add by experience. And even the French word expérience means experiment, right? So it's a transformative battle because Galileo kind of, even though he was in house arrest, he kind of made it out, and now there's a statue of him in several churches around Italy. But it was really how knowledge develops, right? Now, the other battle, of course, is between logic and science, because most people would like to create what's called formal systems. And that means, like chess, that you have everything is known about a system. It makes us humans so happy because if everything is already possibly known, we can just figure out the logic, yes and no, right? And computers kind of are based on those ideas. But biology and science, and I think even more than physics, biology, have observations and things can change, right? Remember, Darwin says that everything changes, that's evolution. And Malthus, who was actually publishing almost 60 years before Darwin, outlined something called um, population principle. So you have two standard deviations, you have the Gaussian curve. So Darwin didn't exist in a vacuum. People were thinking about the world, how to explain it. Malthus was actually an economist who studied poverty. But Darwin must, and I actually know exactly where he lived when I was in Cambridge, but Darwin was surrounded by people who were thinking like this. So if there are populations and there are statistics and there are standard deviations, what is sort of at the core of all this variation, right? So Darwin was in a collective system of thought, as we say. And Darwin, thankfully, could think outside of the box. He was informal in the way that his observations informed the way he thought, right? But this is a whole different subject. We can read several books about this, but I think the essence is sort of what I've said here. The informal system is one that requires new information, and so it will have new logic based on new observations, right? If the Earth is round, etc. Right? But I want to make this a little bit more relevant to our current time. You may not know who Billy Dunn is, but I do. He's the main spokesperson for the FDA for the latest decision to accept aducanumab, the antibody to amyloid beta. And as you know, many of us think this is the worst decisions the Food and Drug Administration have ever taken, because clinical trials showed that removal of amyloid, which this drug does, did not improve the cognition of any single patient. In fact, in many cases, it made them worse. But Billy Dunn, who is more like Aristotle, says, well, it seems reasonable or logical, he says, that if you remove amyloid, the patient should get better. And so the FDA, long story short, is driven by this, and Biogen, of course, manipulated their data and the arguments. But essentially, Aristotle is back, right? If the Food and Drug Administration can be turned over by logic rather than data, we constantly have this transformative battle, right? So, I feel pretty bad about it, but at the same time, this is something that is probably not worse than the worst thing in history. But you can see that people's way of thinking and not understanding how they can slip into these fallacies can create lots of problems. So now we'll see what happens here, but there's an uproar and many people will simply not prescribe this drug, which is shown not to work. It removes amyloid, but maybe amyloid is irrelevant to what they're trying to treat. So what I'm getting to is that when we do science as an experimental design, we agree that belief versus belief is not okay. You can't just argue that I believe something and therefore somebody else believes something. That's okay. It's okay to do that outside of science, but not in science. Data and experiments informs what is reasonable. And finally, a couple of notes about humility. Genius is fallible. So everyone who is fallible is therefore a genius, potentially. So the understanding that knowledge always change and that scientific theories are only as good as what they predict means that humility is one of the most important behaviors in science. It's important in every other aspect of life too. And fools are always wrong except when they're right. So you'll find in science that there is the same battle between good and true knowledge generated by good science, but the interpretations may lead to all sorts of fallacies. But in any case, what we try to do in the lab, right, is to test the validity, veracity of scientific question. And it's really a human behavior or approach or a value system, is to examine biology by controlled observation rather than just leaving it to guesswork. So what we learn, and particularly the style we have in the NRI, is to frame questions rather than talking about this more complicated world of theory and hypothesis and ultimately experimental design. To really give you some words that you may not have heard before, it's the way I'm trying to say that in simple terms is what's also known as a heuristic effort. The heuristic effort is asking the right questions to get an answer. I think it's an old Greek practice. And the data you obtain allows you to interpret. That's what we call the discussion of a paper. And that's known as hermeneutics. So there are philosophers who just study that. And I think some of them can get lost in their own logic there. But for us who are data scientists and driven to do experiments, it's useful to know what the art of all this is. So when you have something that predicts something about the world, you have an hypothesis. You test the data to obtain data. And in practical terms in the lab, we always have to find the test. And this is something I wanted to get students to hear, and techniques to obtain the data. And those techniques and tests, they change with time, right? So classically, my teacher would say, you get a better machine or a better tool, you make better experiments, which is part of the truth, I think. If you can outthink a problem and using this available tools and still get the answer, you don't need to necessarily invent a bigger telescope. The other thing that's very important in experimental design is, of course, to understand that you're planning and you plan to execute something called now rigorous experiments. It essentially means that your experiments should have outcomes that are really reliable. So if you understand that or not, the main goal of experiments, right, is to include sufficient data and sufficient tests to reach meaningful conclusions. So if you see the title above, that means that the data can be interpreted and to answer all of part of the hypothesis. And so it's quite simple in other words, you frame a question, you build experiments, and you get data. But before we move on, I just want to say something on theory, hypothesis, and concepts. The other thing that you will see even when we give examples at the end is that you have to examine your concept and assumptions. This is the same thing as lawyers do, for example, right? So what you do in a classic hypothesis, this is what Karl Popper wrote about, and I am not actually a big fan of him. He wrote something called the logic of Forschung, right? The Forschung means research in German. And he tried to create a formal system around how you could really do this logically. And as I've told you before, I think most of science is informal because you don't know all the data. But essentially what you create is a test of a hypothesis will either nullify it, that and say there's no relationship or dependency between the variables I study and the outcome. So I have shown that there's no dependency. This we call P equals something, right? Back to Malthus. It's something to do with probability and statistics, right? And a lot of people can make this extremely complicated, but if you know what it is, you know what it is. Dependency here means something is causally or at least associated in some way. Could be indirect or direct. And of course, the most important thing with humility is that theory is not reality, but it's hopefully darn close. And so theory should help. You should know enough, even from your own work, to outline what the reasoning is for your hypothesis. So in practical terms, then, what we do in the lab is to frame central questions, as you hear all the time, to find the most effective way of examining those, right? So to summarize, a good experimental design always gives you enough data to know the answers to part of the questions or all of it. A good experimental design, when executed, generates therefore sufficient data when fully analyzed, not midstream. And even if these results go against our original ideas and biases, everybody has biases, they can be interpreted, remember, to report publicly, and that's always been done in science. People write to each other, even letters, the result, and thereby generating new and needed knowledge, which becomes sort of the current belief system, right? And just to be a bit serious for a second, This is actually a professional standard. That's why I joke that this is a 1-hour PhD course for experimental design. And if you're a professional scientist, you have a PhD or else, you have to know these principles. So now back to something quite visual that you see all the time, right? So what we do in the lab is to have a central question, experimental design, experiments, and data, and that feeds back to the central question. And this is what you see every day. Now, experimental design also, I feel, and I really think this has helped me in my work, and I think I published something like 350 papers, all were designed, right? So, intrinsic, sometimes complicated experiments. But experimental design, I feel, should be developed into pilots and then full experiments, right? So the pilot experiment should not be explorations of the hypothesis. You have to hold back your desire there and be disciplined, but generate something technical. And that then feeds, as you can see, into experimental design. Again, the same flow. Raw data is a great term. Don't mess with raw data. Data can never be manipulated, although, of course, with imaging and so on, it's very hard to say what's raw or not, but it's a raw data generates results, which then is analyzed raw data, which leads to interpretations and then conclusion. Now, I wanted to say something that you may have heard also sort of flying around either our institute or elsewhere. That's, of course, a pilot design. And I like it, and Penny too, when we do experiments that are methodological for pilot, because you can be so misled by small experiments. Gating experiments is something that you've heard and we'll talk about. And then a small series versus a full series of experiments. You may hear about killer experiments. It's an interesting term, but there is a certain regression in testing new ideas and new knowledge. And killer experiments are sometimes, I think, almost chosen by whatever you think it means. But it's really trying to limit your experiments to the most critical ones, so that you don't just do descriptive experiments. So you hear often when people are criticizing an experiment or a paper that it was descriptive versus mechanistic, right? But the killer experiments kind of try to nullify again, or find the most critical test fast, right? Or in industry, they would say fail early, fail fast. But I think it's a little bit of a bravado kind of style. So the other thing that's taken me a long time to understand and teach is to see why there is so much trouble understanding what controls are. So in the logic of an experiment, people will always say, well, it has to have the appropriate controls. And you can find that particularly younger scientists will come up with lots of controls. But the question I always found was that to ask is, what are you controlling for? What is the new question you have? Are you controlling for whether 6-hydroxydopamine kills dopamine neurons, or whether AAV can generate a gene delivery? Meaning you have to really limit yourself, because of what we call resources in the end, to understanding what you're controlling for. And back to design, if you know your central question, you can then deduce what are the most important controls I want. What do I predict is something can be very hard to do, and I try not to predict too much, but rather design against whatever outcome I have. It either kills the bias or the idea I have or we have, and so on and so forth. But that's something that I think is very hard to teach, but in a refined way. What we're controlling for can really streamline. I think people come to me for help on that. What is the central thing? Now, what we're not going to discuss today is statistical principles and power analysis, but that's something at the undergraduate level I doubt you've seen, but that's something that comes with experience that leads up to a PhD. If you don't understand statistical principles, you're truly lost, but I think most people are taught that at an early age these days. Power analysis is really saying, how many observations do I need to make, given the variation in my sample, to obtain statistical significance for the hypothesis I'm testing. And then a little bit more sort of entertainment here is experimental design meets reality. And even though we make these designs, they are frequently flawed. In fact, you can say all of the designs are flawed. But it's important to be alert to not just surprises, but that methods can fail or that methods improve. That's why we're so eager to learn about what other people do, right? Or what our colleagues do, go to meetings. Because with a little bit of improvement in a method, you can see things or learn things that other people haven't seen. We're back to Galileo again, right? He had this telescope. and the cardinals did not. So that's one thing, that adaptation is very important and learning new techniques are, but only, I feel and think, when they serve the purpose of solving a problem. Just learning techniques, I find it's not something that interests me, but it could be that other people find that their skill set is there. Now, even more advanced is to understand context, situation, method, techniques, and resources. And that's something that we kind of almost have to deal with on a specific case. But what I mean by that is that you have to think all the way from which laboratory you're in, right? What methods are available, but also what resources are available. Who can you work with? And for those of us who write grants, it's a constant struggle to have enough money to do an experiment. But sometimes, even with lacking resources, you can outthink the design, you can trash the design and redesign and figure out a way, a little bit like a practical person, how you can answer part of the questions without spending millions of dollars. And then the other thing that comes with experience, I think, which you can't kind of avoid, it's a bit like people betting on the stock market. It's genuine exploration actually takes a fairly determined approach, right? And so you can find people, and here are allegories, right? What's the story? A long and winding road. It looks like when you design the first experiments, it's going to be straightforward, but frequently you're going to run into these problems that I mentioned as adaptations, right? And the tortoise and the hare, if you do a really good design and it takes time, you may actually outrun the competition or people who are testing the same hypothesis because they're going to take shortcuts. They rely on experiments that are not strong enough to withhold the test, right? And sometimes, therefore, it takes years to do this work. And believe me, if you're an explorer by nature, you have to then build in patience, right? Whether you use racers, cars, bikes, or simply walk to the answer. But it's really the experimental design's help to structure your path, right? So in short, if you have good behavior, and that's something I would like to expand. Most people talk about design as an abstraction. I actually think, from my experience, that scientists have behaviors like every other profession. And there are good behaviors and, frankly, less successful behaviors. So when you work with scientists, we're usually a global profession. It can be any country, anywhere. But you can see who sort of blends this idea and understanding of all these things I talked about and come up with design. So then, so I told Penny about this idea, and we said at the end, heck, we got to show something that's not just sort of talking to, for those of you who have an ability to understand these concepts up front. But what kind of designs have we done? So Penny and I discussed this for a while, and we came up with, and Penny searched the entire databases. We've had computer systems here for decades, and we looked up some really, really old designs and then some new ones, right? But so some of these things are so old that I had to really look at them for a long time to remember all the pieces. But just to give you a little personal vignette here, I remember thinking through a theory when I was first a graduate student, but then later when I became an assistant professor. And I summarized it in an article which was really just 3 pages in TINS in 1993. It was called On Neuronal Health. I was fascinated by how cells can exist sort of in between health and disease, right? And I've done some discoveries early on in the late 80s and 90s where I realized cells were not dead, they were just injured. And all of my colleagues, and there were a few of us who worked on this, we realized that people, usually pathologists, had written off cells that they were dead because they couldn't see them. Well, we use some techniques to illustrate something called independent labeling, so retrograde anti-red tracers. We can watch neurons, we damage them. They lost all their transmitter enzymes, but they were not dead. And in the peripheral nervous system, they recovered, right? So I worked on this idea that cells could exist between these states of very, like an equilibrium. And then when they get up to a certain threshold, which we wrote about not a few years ago too, they can pass an irreversible threshold. And I was called living in the collective time of the 1990s, where people were not so sophisticated about this. And so now it seems obvious, but we had to argue and battle these positions, meaning we wanted more evidence about where the cell was in this equilibrium. And I predicted in the theory that if you have 2 subthreshold injuries, they could add up to a bigger injury. And I remember talking to a very famous scientist who was a little bit obnoxious, and he said, well, that should work, but I don't think so. And I said, well, where's the experimental evidence? And he said, I don't know. I provided the evidence in something called Simpson and Isacson, where we had one dose of a toxin that didn't give any damage, and then another dose, actually a complex 2 inhibitor that didn't damage the brain too. I was like working on Huntington's disease in my thesis. And when we combined them, we could, create large lesions. And so this idea of testing neuron health, fed a lot of the experiments in the lab at the time. And we were always been a happy bunch, right? But back to a long time later, around 2005 or so, we had now worked out many of these we worked on, heat shock proteins in the 10 years before, and a lot of other things. But here was something that really was unique to our lab, and nobody ever had done these experiments before. So here, the idea was that inflammation or triggering inflammation would create this sub-threshold status, which then would, with a little bit of injury, would create a bigger lesion. And my inspiration for that was actually listening, and this is the truth, the pandemic in 1918-20. And I remember going to meetings and not fully, or my curiosity was not satisfied by the answers and the ignorance that was displayed around me. People said, oh, we don't know. And who knows? But I said, so that many cases occurred with Parkinson's disease, and yeah, but we don't believe it has anything to do with inflammation anymore, said clinicians usually. And they had something at the time. Already amyloid was, can you believe it, getting hot. And there was a few people who talked about synapse degeneration, but they were usually ignored, right? So collective thinking is very dominant, right? So, well, anyway, nobody argued with this. So in the lab, there was an interest, and there was a postdoc who came around that time, 2006, 2007, and we discussed this idea. There was one experiment that had been done in the gut where they used these various substances, but to induce inflammation, and that had some effect on brain development, a colleague of mine. So we decided from scratch to try some of these things. And the idea was to create inflammation in the substantia nigra that didn't kill the neurons, and then see if that exacerbated. And the hypothesis was that if the inflammation comes first, and then you provide a mild injury, that would be worse than providing a mild injury followed by inflammation, because that's how the pandemic worked, right? So here, Penny found this design from a long time ago, where we did at first the pilot experiment. We had an inflammation by very low dose of LPS, we know it's the TOLAC4 receptor, over the substantia nigra. And then we also had, this is sort of half the dose or a third of the dose of this 6-hydroxydopamine injected into the striatum. And then we waited 12 days. Why 12 days? That's the thing I didn't tell about experimental design, because I had studied this degeneration for years. If you do a lesion in the target zone with 6-hydroxydopamine in the striatum, it takes about 21 days for the cells to degenerate. And I noted in many of those experiments, there was a rapid phase, and it took about 12 days, but then there was persistent, long degeneration that occurred around, from at least we had data points at 12, it may occur, as you know, at 9 days, but it was a curve and a slow decline. And my intuition, my hunches, based on years of observations, was that this is a different phase. I thought it was inflammatory. In fact, guy in Argentina, Pitosi, had the same idea for different reasons. But anyway, so this experiment, the pilot generated a cytokine profile, which we then used extensively. We didn't develop the method ourselves. We sent it out and had it analyzed. But here you see the interleukin, interleukin 1 beta came up, for example, and other things. We also fixed tissue, like you see, we studied a number of things and looked at these various outcomes. So that pilot experiments gave us hope for what to develop. And this is not the final design, but you can see here that the design says if you do inflammation first or lesion first, and then followed by here on the left side an injury, or then small lesion and then followed by inflammation, then we wait a certain amount of time, 21 days, we tested things, we made counts, and this is not the final, but you can see here that we were already thinking about what we have to do if the experiments didn't work, right? This is not in the publication, but we would have to adjust parameters. We may have to change lesions. We have to do other things. Just to illustrate that we were working very hard here, right? Further tissue analysis, we thought about cytokines. Maybe we have to look at microglia and astrocytes again. But thankfully, the first experiment worked, right? But just to illustrate, so here you go. So 2 or 3 years later, we had a complete study, and it was really earth-shattering for us. I think in a way, this paper, if fully analyzed, would be one of the first to develop this idea. But we also were able to block the degeneration by interleukin-1 receptor antagonist, which I think was probably 5 to 10, if we can be proud here, but 5 to 10 years before really people realized what we were doing. Thankfully, Fernando Bitosi in Buenos Aires were doing similar experiments. So there was at least two of us, or two labs, I should say, who had that going. But you see, that's sort of, if you go to that paper, right, in Journal of Neuroinflammation, you can see that the design in the end were slightly streamlined. Here's another one that's a little bit more recent. And this is the work on lipids. So there are several papers that are theoretical that follow sort of this in nature. We had an overview on neuroinflammation that Michele Delady and I wrote that was published in Science Translational Medicine, that outlined the overall theory about how inflammation can trigger degeneration. But at the same time, the clinical data from the world of glucocerebrosidase really took many of us by surprise. So we didn't discover that. It was a clinical discovery. But our team started working very quickly as pioneers in this field. And obviously, it was very competitive, as Penny and I can tell you. But we really had a genuine interest for this system, right? So we wanted to look at the lipid induction. And also, we are a translational lab. We work for therapeutics, right? We just don't do this to study the stars in the universe, but really to see if we can help people by these discoveries. So here's the design. First, we had to do a pilot experiment to decide which AAV serotype would have the best diffusion. We had used AAV22 before very successfully. In fact, this pilot experiment may or may not have been made a difference, but we looked at 22, which doesn't diffuse much. And we had used that quite effectively to limit the injection to the Sancionaga pars compacta. But we tested various things. And as you can see here, the virus titers had to be determined, the flow rate, the gauge of the needle. These are the hard things that scientists do, particularly the exploratory scientists. But you have to figure out your techniques, right? We have different coordinate sites. For those who've done poly-IC, you remember what it takes. We had all the tests that required that to work out, and so on. And we worked in rats, which is a better animal to do behavior in, but we changed around. So this was actually quite important, and we decided to use, I believe, the AAV25. So then this is again something that we pulled off the computer or that Penny find, or even her own files. And you can see here very much what it takes, right? We had to generate the AAV and the timeline. We had to do these pilot experiments, and then we started with the intracranial injection, and then we had a plan here. The final experimental design, the ASO Thy1 is an overexpressive mouse, a bit of a monster mouse with massive amounts of alpha-synuclein. And then we had the plan based on our pilot, right? We had behaviors, and then we perfused the animals and studied their activity. And this led to publishable work, right? So we felt like we'd done a good job. And this was one of several papers, there were 3 even in that 1 year. But we managed to show that this gene therapy prevented alpha-synucleinopathy of the midbrain dopamine neurons. And I think we're very proud of this paper because it's quite real in the sense that it has both description and discovery in it. And it was followed by other papers. But again, if we have an internal assessment here of what we did, it was actually to some extent ahead of the other teams because they kind of went through studying the Gaucher's model. We went straight for the alpha-synucleinopathy. Not that that's going to help people develop. Now people are trying to, again, reduce alpha-synuclein rather than prevent degeneration, but that's another story. But this is sort of how you see this result.